Tessa, and thank you, JP Morgan, for having us, and I appreciate everybody in the audience. And as I'm sure everybody recognizes, we'll make a few forward-looking statements. So please do refer to our 10Q. We spent a bunch of time writing our risk factors. So 2024, I think, really promises to be an exciting and potentially, hopefully, transformative year for the company. We spent a lot of 2023 preparing ourselves for a bunch of clinical data. And at this time last year, we had no programs in clinical testing. Today, we have 4 across 7 indications. We'll treat something like 40 to 60 patients in clinical studies this year. We'll have data available from every one of them. And during this talk, I'm going to give you a little bit of information that we haven't shared before on why we're so optimistic about where this will take us. We've really focused the company's effort in the near term around a platform of overcoming allogeneic rejection of cells. Meaning you put my cells in you, your body's going to reject them. And hopefully, with the technology we have and the progress we've made, we can transplant cells more broadly. And we're doing this in 3 different broad therapeutic areas today in the clinic. One is in B-cell cancers like lymphoma and leukemia. The second is in B-cell mediated autoimmune disorders, lupus nephritis, encapsulitis, and a whole host of others over time. And finally, in type 1 diabetes. We have a number of drugs coming behind that, which we'll tell you about some other time. And hopefully we have the balance sheet to allow us to really read out all that we're doing. So this is just a little bit of more detail around our portfolio. The lead drug, or the first drug into human testing, is a drug called SC291. It's a hypoimmune modified, which we're going to talk about in a second, allogeneic CAR T cell targeting CD19. And that trial has been up and going, and we'll show you a little bit of data from it. Just last week, we had a drug called SC262 allowed by the FDA. It targets CD19. It's a CAR T cell targeting CD19. It's for patients who failed a CD19-directed therapy previously, a big unmet need, and someplace we expect to move quickly. Over the course of the last 18 months, there's been a lot of excitement in the field, which we'll get into, around the potential to even have curative intent with autoimmune disorders. And we have a drug, SC291, that we're taking forward in a number of these disorders. The IND was allowed in the 4th quarter, and again, you can expect to see data later this year. And finally, we're moving forward in type 1 diabetes as we speak, and we'll get into that. So a little bit around the problem. Since the advent of transplant medicine, the rejection of allogeneic cells has really been a big part of the limitation around broad implementation, and the same is true as we move into cellular therapy. Most have tried to get around it by substantial immunosuppression. That both limits the number of patients who can do this, but has a lot of long-term toxicities. The field's tried to move into autologous cells. They work in some cases. There are only a handful of cells where that's possible, and scaling it has been complicated. And all of the efforts to really gene-modify allogeneic cells haven't been complete and haven't worked to date. So what's our special sauce? If you take a step back, there are 2 big arms of the immune system. There's the adaptive immune system of B and T cells. Dealing with that has been relatively straightforward and known how to do for a while, and knocking out class 1 and class 2 MHC. Unfortunately, or fortunately, tumors and viruses figured that out a long time ago. And so we've evolved the innate immune system, and particularly natural killer cells. And so overcoming the rejection of natural killer cells has been the real challenge of the field. And what we've discovered, our team has discovered, is that overexpression of CD47 in the context of knocking out class 1 and class 2 gives you the potential to completely evade immune detection, transplant cells with no immunosuppression, and hopefully have them live as if they were the patient's own cells. So we've done a lot of work in the preclinical setting. And I'd go so far as to say we've proven that we can overcome allogeneic rejection in mice, humanized mice, non-human primates, amongst others. And these data are published across multiple journals, including things like Science and Nature journals over the course of the last 12 months. So take a look if you want to go more deeply. On the right-hand side, what you see actually is a transplantation into a non-human primate of a pancreatic islet cell. challenges. And the goal of doing transplants of pancreatic islet cells is to overcome type 1 diabetes. Our team wanted to do one more experiment as we were waiting for humans, and I want to share that with you. And the goal here is to figure out, can you create type 1 diabetes in a non-human primate? We've never shared these data. Transplant allogeneic cells with no immunosuppression, and normalize the outcome for the non-human primate. So that's the goal. Can we survive and function? Will we get long-term glucose normalization? And actually, at the end of it all, can we eliminate these cells? Which is both, we could do that to validate both our mechanism and a safety switch. So this is where the experiment work. And this is a look at the animal's glucose in their blood. And the green is normal, the white's a little abnormal for first meals, and red's abnormal. So give the drug streptococcus STZ, and that eliminates the pancreatic beta cell. And you can see the sugars are in the animal are all over the map. Over the course of the next 2 months, we were able to stabilize the animal with insulin, and at that point, the veterinarians allowed us to try to transplant allogeneic cells with no immunosuppression. And what you see is for 6 months, the animal remained euglycemic, off insulin, with no immunosuppression. It's a spectacular outcome. The animal was able to, at that point, reinstitute itself in with the colony around it and was thriving. We decided, though, that there is some risk that maybe the pancreas had regenerated. And so our mechanism of overexpressing CD47, we can give an antibody to that and see what happens. So what you'll see is when we gave the antibody to CD47, the beta cells were killed, and the animal became diabetic again. And on the right-hand side is what you see is the C-peptide, the measurement of insulin, which goes right along with that functional outcome. So a great outcome, and about as all you can do now non-clinically. So it's time for us to move into people. As I mentioned earlier, we're going after big markets and large unmet needs. Blood cancers, where over 100,000 people per year die in the U.S. and Europe alone. B-cell mediated autoimmune diseases, where you have over 5 million patients suffering, and type 1 diabetes, which is a global endemic and something where we think we can have a large impact. I'll start with blood cancers, and I think most people here recognize that with the advent of autologous CAR T cells, there's been the possibility of curative intent in treating patients. And we've also seen a number of companies try to really scale the capability with allogeneic CAR T cells. The challenge with the autologous has been just patient access in many regards. And despite the fact that 100,000 people die per year, what you see here is this yellow circle is the number of people who get them per year, around 11,000. And unfortunately, the light blue is the number who benefit from them. So there's still a very large unmet need. Allogeneic CAR T approaches to date have shown that you can get a pretty good early response, but unfortunately, as the patient's immune system recovers and reconstitutes, the cells are killed, and they're not around long enough to really lead to a durable, complete response. So we're moving forward with a drug called SC291, and what we've done here is we've gene-modified donor-derived CAR T cells using Cas12b, and you see we knock out 3 genes. We disrupt MHC class 1, MHC class 2, and then we stop TCR function. I can get into why in a minute. We then overexpress the CAR and express the CAR and we overexpress CD47. And so that's what we do. The real goal is to figure out, do we have a drug here? And the first part of understanding do you have a drug is, can we overcome allogeneic immune rejection? If we can do that, that's probably 80% of the risk of the program and 80% of the risk of the company, right? That is something that we've seen in non-human studies that we are really quite good at. We want to make sure that the animal data translate into people. If they do, can we make great CAR T cells? And that's measured by safety and efficacy of the program early. And then we want to see how does that translate over time into durable, complete responses. And finally, we need to make sure that we have a real scaled manufacturing process. So these are the data for the first time that we've shared. We've enrolled 6 patients. We enroll about 1 a month. That's just what you have to do during this time. And on the right-hand side, what you see is that, importantly, the drug has been very well tolerated. It's a dose escalation study. We started at dose level 1 at 60 million cells. We're now at 120 million. Next will be 200. We've had no dose-limiting toxicities. In fact, we've also had not even any grade 1 CRS or ICANS. What you can see is that 3 of the 4 patients have had at least a partial response, even at these low doses, and 2 of the 4 patients are in an ongoing complete response. So the drug is working as we hoped it would on the safety and efficacy side. Stay tuned. We'll have a lot more data as we get through dose escalation. We're able to enroll patients and treat patients very rapidly, and we'll have a lot more as the year goes on. But as we go through that, the real key question is, how does that early data that we've seen in animals translate into people? And one of the bugs of the drug is that when we take donor-derived T cells and we gene edit them, we're really good at it, but we're not perfect. And so the drug is a little bit of a gamish. You have all of the hip CAR T cells, which have every single gene edit we want to make. But we also have some cells that just express CAR. They haven't knocked out class 1 and class 2. And we have some cells where we've knocked out the class 1 and class 2 MHC, but we don't overexpress CD47. And what's been seen in the field to date is when patients get an allogeneic CAR T cell, they first get lymphodepletion, chemotherapy, and as their immune system reconstitutes, the CAR T cell is killed, limiting efficacy. So what we want to see here is what we on the right-hand side outline, is that as the patient's immune system reconstitutes, our fully hypoimmune cells live. And we'll know the immune system is reconstituted because you will generate an immune response against the partially edited cells. So this is the test of, does this really work in humans? So what we see is what we hope we would see. I'll start with that. On the left-hand side, what you see is in patients who don't knock out their class 1 and class 2 MHC, or sorry, not in patients, in cells that aren't knocked out. And this is real patient blood, right? They generate T cells over the course of several weeks that will recognize and kill the allogeneic CAR T cell, non-gene modified. What you see in the middle panel is if they knock out class 1 and class 2, as you'd expect, T cells don't recognize these cells. And what you see is our fully gene-modified cells. Again, there's no T cell response, T cell recognition to them, and they can live. So next question is antibodies. So what do B cells do? Again, patients will generate an antibody response to cells where you have an intact class 1 and class 2 MHC expressed. But they will not if you've knocked it out, and they do not to the to our fully gene-edited cells. The field has seen this broadly. So you've seen a number of companies that have knocked out class 1 and class 2, but the drugs haven't worked as hoped. Because the key has been, as we mentioned, natural killer cells. So this next slide is, in many regards, my favorite slide in the whole presentation. It's the kind of the killer slide. It's the picture of what happens. So this is actual. These are NK cells from patients 28 days after being treated. And what you see is when you have double knockout cells. This is what a lot of the field has done with class 1 and class 2. The green cell is the CAR T cell, and the rest is the NK cell. And very rapidly, the NK cells will recognize and eat and destroy the CAR T cell. We're often asked, well, what happens if you knock out class 1 and class 2 and overexpress HLA-E? So we contrive that system for you. And again, the patient's NK cells will rapidly recognize and destroy those cells. And what you see here on the right-hand side is what happens to the NK cells and our fully modified CAR T cells. And they bump up against it, but there's absolutely no killing. And this is the most important slide in many regards, because it tells you that with real patient blood, real patient samples, we've overcome the large challenge in the field, which is NK cell recognition of these cells. And just to be sure, we said, hey, what if there's something else in the blood that might kill these cells? And what you see is, against wild-type CAR T cells, as expected, they're dead. Again, that's just an allogeneic cells, my cells into you. If you knock out class 1 and class 2, the blood from the patient will kill those cells. But what you see on the right-hand side is our fully gene-modified cells. They really do work. So the drug is working exactly as we hoped it would. And the immune evasion that we saw in the preclinical setting seems to be translating into people. So stay tuned. The early data suggests that we have the safety, the desired immune evasion, and the clinical impact that we're hoping to see. We'll have a lot more data as the year go forward, and we'll share it across all of these different parameters. So We're unlikely to do it at another investor conference. It will most likely be at medical meetings, so stay tuned. I want to move next to autoimmune disorders. And over the course of the last 18 months or so, the field has really begun to understand the potential of CAR T cells in this space. That being said, if you go back over the last several decades, people have been targeting B cells with autoimmune disorders with a number of different drugs, and there are around 70 different indications today where at some level there is evidence that depleting B cells or knocking down B cells will have a clinical impact. Unfortunately, with the antibodies, what you see in the middle point is they're really pretty good at knocking them down in the blood, but they're unable to get to tissue-resident germinal centers. And that's where a lot of the problematic B cells reside. And what we know is that the depth of B cell depletion across multiple studies really does predict the efficacy in all these diseases.